Hello, everyone. Welcome to the seminar today. The talk of today's seminar is Bioinformatics for Medical Laboratory Technologies. I'm Dr. Tian. I'm the Program Director of HKU Space. So today's topic, I would discuss why bioinformatics is important. And I would also take this opportunity to introduce one of our new program at Space, which is bioinformatics for MLTs. So let's begin with some background. Why we need to talk about bioinformatics now, and then why it's important for the medical laboratory technologies. And then later I would give more details of our new program. So the reason why we need to talk about bioinformatics, we have to talk about technology first. So with the development of new technologies nowadays, the molecular tests or the genomic tests produce a large amount of data from patients or from health individuals. And moreover, nowadays the diagnosis tend to called precision medicine. So we try to find the association between the genomic data and the disease. So this new technology produces a large amount of data, especially the genomic data. So that need data analysis to uncover the clinical significance behind. Well, we can take human genome as an example. Actually, for the whole genome of human, there are more than 3 GB data produced just for one cover. Well, for some of you, if you know a bit of sequencing technology, then you probably know if we sequence the genome, we can't just cover it once. We need to have a sequence depth of maybe 10, 20, or 30. So you could consider if we cover it 10 times, then it generates 30 GB of data. Well, if we cover 30 times, then that's definitely about that's generate about 100 gigabytes of data for one human genome. So this produces a large amount of data. Well, moreover, I think you may understand that not all disease is linked to one single gene. Some disease is caused by multiple genetic disorders. So that also requires us to analyze the relation, the association among different genes. So if you see the figure here, this demonstrates the human genome. So we know human genome consists of 22 pairs of chromosome plus XX or XY. So if we need to study the association or relation between different sites, then it produces even large amount of data. Then obviously it's not possible for us to analyze such data by human, by manually. So bioinformatics, well, from the name, you could know that bioinformatics is an interdisciplinary field of biology and computational science. So basically, we use computer programs to compare and analyze the data generated from biological or biomedical field. In general, I think nowadays we mainly focus on the analysis of DNA data or RNA data. So those generated from sequencing. Well, here I took the very well-known example, which is the COVID. So we know the virus of COVID is SARS-CoV-2. And then it has many variations. I think we all heard that alpha, beta, and nowadays it's Omicron, and then BA1, BA2, BA4, BA5, and so on. So how to analyze such change in the genome of the virus, how to compare or how to study the association between different strains of this virus. It's, again, not possible by doing analysis manually, because although the genome of this virus is relatively small compared to human genome, which is only 30 kb, but if you have different strains, different variants, then we'll do the comparison. And we want to study the association between different group, then definitely you can't do it on your own. So the figure shows I cite from one paper, which is an early paper to study the association between different COVID variants. Then you can see to draw such we call it phylogenetic tree. It's a nice tree, and then clearly showed connections, show the association between different group, different cases. So this gives advantage for doctors, for healthcare professionals to understand this virus. So definitely this figure is generated by bioinformatics tool after the analysis. So that's why we can find that bioinformatics is very important in biomedical studies. So, okay, the next one is why we focus on medical laboratory technologies. So we know that MLTs are those professions who are registered to practice of processing clinical or medical samples, especially the human sample or animal samples. So the current training, especially the undergrad training that in space, we have a higher certificate in medical laboratory science. We mainly focus on the clinical chemistry, hematology, histopathology, and microbiology. Well, we are also includes some of the new technology, those genomic technologies. But again, the traditional area is still the major learning content for these programs. So it's not yet included in the training of the bioinformatics is not yet included in the training for MLTs. However, we know that in Hong Kong, the Hong Kong government has proposed already that the strategic development of genomic medicine, and then the hospital authority it also has a direction to provide genetic and genomic services. So we can see that there's a need for nurturing the skilled and competent workforce in HA or in other labs to produce this data and to analyze this data. So we know once the genetic test has been performed, then as I explained in the previous slide, there would be a large number of data generated, and definitely we need some professionals to analyze such data. So there's a need of enhancing the knowledge and competence of MLTs in analyzing the sequencing data, let's say all those DNA and RNA data. So we propose a new program which has been approved in space. This would be a postgraduate certificate in bioinformatics for medical laboratory technologies. You may notice that, well, in Hong Kong, there are some courses related to bioinformatics, like the University of Hong Kong and the Chinese University of Hong Kong all have bioinformatics at undergraduate level or postgraduate level. However, those programs normally focus on the full training for bioinformatics, including the principle of computer science, and then as well as the later parts which may focus on research. Well, that's a full training. I would say those are very good programs in Hong Kong. However, for our program, this program focused on the application of informatic tools in in analysis and interpretation of clinical data. So basically, these programs would give more time on the application. But of course, we will cover some basic principle in computer science in those genetic information, but that's not the point. We want the student, after they complete this course, could use the program, could use the analysis tool in their workplace, which also focus on the clinical data rather than research all those common data. Again, similar to most of our other programs, this program will be delivered in part-time mode, and the students are expected to complete the whole program in 9 months. So normally it's more flexible for the working people, especially for the MLT. We know that it's quite busy in the lab, so it's very good. It's a very good opportunity to study this program in part-time. Well, the later part, I would cover more details of our program. This program, I would introduce about the intended learning outcomes, and then the details of each module, so you know what is expected to study in this program, and then the award and what resources we have to support your study. And then most students may be very interested about the requirement to apply this program. So those content will be covered in the following slide. Well, what we will learn from this program, we would have 4 learning outcomes. Students would be able to explain the principle of genomic technologies, and students can achieve genomic sequence by using appropriate database and software. Students would be able to develop computer script to write some programs to complete fundamental tasks on data processing. And then students will be able to apply those tools to analyze the genomic data for clinical diagnosis. So our aim is, it's very straightforward. You understand the technology, and then you can use the existing database and software to do the preliminary comparison and analysis. And then you will be able to write some very simple script, or maybe revise or modify the existing program to help you to fit your lab's needs. And then the last one, which is the goal of this program, to apply the tools for data analysis. So in this program, there would be 4 modules. The first module is genetic and genomic technology. And then we have an introduction to the bioinformatics. Then the third module is programming for biological data. And the last module is the application. So I would explain each module one by one. Well, this program is planned to be launched in October, and then we have been approved, just approved by our board. So please check our website. We would update the information soon, so you will know how to apply to this program. Well, the first module, genetics and genomic technology. So from this module, we would help the student to review the basic knowledge in molecular genetics, and then also help the student to understand the principle in molecular technologies. This module covers lecture, laboratory practicals, and examination. So it's a 9-credit module. In this module, the student will also have some assessments of essay and the written examination. So the design of this module is that before we use bioinformatics to analyze the data, we have to understand where the data came from and then what's the meaning of the data. So we try to have a very simple review session for you to review the knowledge in genetics, genomics. Those basic knowledge would be a very very fast session. And then after that, we would cover different technologies in molecular medicine. So those technology is different platform for sequencing for different array, and then also the technologies, the technology behind. So the reason why we try to cover this is that we are not only use the technology, but we have to understand the rationale behind, so the student will be able to understand where the data are generated. And then once you understand how it's generated, then you will be able to process for the next step for analysis. And then it's also important that for the QA, quality assurance, quality control process. So if there's anything wrong, if you find the data is not good, the data quality is not good, then at least you could trace back and understand where the problem could be. So it's very important to understand such principles. Well, we also cover different sequencing strategy. Either it's a whole genome sequencing or it's exome sequencing, only trying to sequence the exome of target genes, or maybe it's a target sequencing, try to focus on the important region that may link to a disease. So all these strategy link to how the data generated and how the data looks like. So let's go to the detail of this module, the syllabus. Revision of genetics could be the first topic. And then we start from the PCR and Sanger sequencing, which won't generate a large amount of data, but is the basic principle for the new technologies, for the following up next generation sequencing, and also the third generation sequencing. We try to understand different platform. Those platform could be those widely used in local market, as well as the new platform which is developing. So that help that prepare our students not only for the current situation, but also once the new technology is ready, new development is ready, then we will be able to handle all those new technologies. And then array-based methods, like you probably heard about microarray, and then RNA sequencing, target sequencing, whole genome sequencing, and whole exome sequencing. Normally, target sequencing and whole exome sequencing is more widely used in clinical setting, and whole genome sequencing, well, is also used in clinical testing, but I think in research that's also a very important area. The whole genome sequencing probably give more potential to study the unknown relation between a disease and the genomic variants. And we would also cover other omic technologies, proteomics and the metabolomics. So you have a whole view of all the new technology. And lastly, but also a very important topic, is about ethics. So the ethics in using this genomic technology, because we know that in genetics, the data generated is not only linked to one person, it also linked to their family, because basically the genetic information was passed from parents to children. So the data privacy and security is a very sensitive topic under genetics and genomic technology. So we will also cover that, how to get the consent from the patient, and then how to properly handle those data to avoid any legal issues. So that was all for module 1. Well, for module 2, introduction to bioinformatics. So after we understand the technology, how the sequencing data was generated in module 1, then we start to touch something about computer as well as the biological databases. So in this module, we would introduce the computing environment to students. and then also cover the skills of using those widely used genetic database and some software. So this module consists of lectures and tutorials. So if you notice that in most of this module, we include tutorial sessions. So for those experienced tutor could have a chance, so for the student would have a chance to discuss with those experienced tutors, and then they will help you to study because we understand for most of the students, this computer part would be quite new and maybe quite challenging. So we would arrange a lot of tutor for you to help you to understand. And then this module, start from this module, we would focus more about application. So we would have this worksheet and report to assess whether students will be able to use such technologies, such tools. So module 2 would cover those databases, online databases from which you can retrieve the reference sequence, and then you can also get the information from the previous studies. So from like the gene bank, the NCBI gene bank, or the UCSC genome browser, which give you different mark of different annotation for the whole genome. So you can achieve lots of useful data. And then the commonly used software. So those software will help you to do some tasks like do alignment, trying to compare different sequencing results and identify where the mutation is and find the difference. Although I heard that for sequencing result, especially for Sanger sequencing, in some lab, some staff still prefer to use the manual alignment, trying to identify the difference. But definitely once you got more and more data, it's not possible to do it manually. So we would use computer software to help us. And then we also cover a very important point. After module 1, you know where the data generated. In module 2, we would also talk about how the sequencing result looks like, what's the format, data format generated by different technology, and then how to utilize these results for the next step in analysis. Well, the other important point in this module is the computing environment. So computing environment is how to set up the computer to perform those bioinformatic tasks or use those bioinformatic tools. So in this module, we would not only cover the commonly used Windows environment. Of course, Windows is one of the important role because most of the PC are using Windows, but we would also cover Mac OS, those Apple computers, and as well as Linux or Unix system. So student would learn a very basic command line method, so you can handle those. We are not training the computer scientists. So all the tools or all the language we learn here, all the commands we learn here are very fundamental, just help you to handle the files or to set up to run those tasks. So don't worry about this. Of course, we would also cover like the internet, the setting, and then how to get the information, how to get software from the internet. So okay, that's the detailed syllabus in module 2. We would cover the common database and software, and then the data format of sequencing results from different platforms. We would also cover the genome-wide association studies. Well, someone may argue that genome-wide association study may be a bit challenging. Yes, but here we cover introductory level, and then it's very important because we try to link the disease or the phenotype to its genetic variant. So this would be very important. And then next part is about computer environment. So how to set up computers. We would have an introduction to Linux because many common tools are under Linux. So we learn the basic command. And then the last part is the we talk about Git. So for some of you don't understand it, it's very simple. So it just help you to get the software which has been written by other scientists. So how to get the existing software for you to use. It's a very good system. So from this module, you will learn all these basic setting for bioinformatics, so get you ready to do the analysis. Well, next module, module 3 called programming for biological data. This module covers the basic statistics, and then we would also teach you some fundamental programming skills. In this program, we choose R as the programming language, and of course, its application. So why we design the module to cover bioinformatics and programming? Bioinformatics is biostatistics, because statistics is quite important, especially when you analyze the data and then try to identify whether the quality of data is good enough or it needs some further filter to select the useful data. And then it also give you a strong support for you to decide whether the result is reliable or not. So statistics is definitely very important. In this module, we would review the basic principle of statistics, some fundamental concept in statistics, and then how to use the statistical tool in bioinformatics. But don't worry that we won't cover too much detail because we are not training statistician. So it's again how to use those tools, how to interpret the result. At least you have to understand what's the meaning of a p-value, or how once the result give you once those tools give you a result, how to interpret those results. Again, we focus on application rather than the principle or the theory. Well, the second part about programming, we learn some basic programming skills. I think you may consider it similar to the those technology that's although nowadays in the lab a lot of tests, a lot of analysis has been done by machine. It's highly automated. It's a full automation system, but the technicians still need to understand the principle, how it works. So here it's the same. We will use bioinformatics tool for analysis, but we also need to learn the principle, how it works. So we learn a little bit of programming for you to run the program, for you to link different programs together to process the data, for you to maybe even make some modification on the parameters to fit your lab's data. So that's why we need to learn some programming. Well, it takes us some time to decide which programming language is the best. And finally, we choose R as the language to be covered in this program, mainly because it's easy to learn, it's not too difficult, and then it got a lot of existing package. That means the previous scientists already write some good program and then put online for you to use. So for many of the analysis, you don't need to start from beginning. You just need to identify the most appropriate program for your use and then download it and run it. So that's a very good program. And I would say a simple program, and it's very strong in its data processing and statistics. So yeah, as I mentioned, we would review some basic statistics principles. What are those t-tests? How to decide, how to determine the standard deviation and so on. So it won't be very challenging. And then the next step is the R programming. Well, another advantage for R I forgot to mention is that R can produce very nice figure. So for the data interpretation, a nice figure is very useful. So you can really present the results, visualize the results. So easier for students to understand the meaning behind all those data. So okay, the detailed syllabus of this module. This module would cover revision of statistics, biostatistics. So basically, that's the application of statistics in processing biological data. And we have an introduction to R to tell you how to get the R. It's a free software. So how to get R, how to install it, how to run the basic R command. And then we would also have the programming. It's very simple, and we would have experienced tutor to teach you step by step. So don't worry about that. We won't touch the hard parts, just the very fundamental language. And then the last part is to teach you all those existing R package which could be used for bioinformatics or which are widely used for bioinformatics. As an example, so you would be able to get those package and then use what you just learned, the basic R program, to run it, to connect those different scripts to achieve your purpose. So we would have some tests, some lab report for you to complete. So after this module, you will be really able to analyze the data by using computers. Well, the last module, module 4, we focus on applications. So we would invite several experienced teachers who are working in the medical lab, and they share the experience of the application, which covers the tools in different labs. So from this module, you would definitely study how to apply those programs. So here you may see that, okay, I cite some photos from those software. Again, you would be able to use the software to generate nice results, and then for you to interpret the trend, the distribution, the proportion of different components, and then also to analyze the sequencing, the sequence among different species or different individuals to draw a nice figure. So that's the ultimate purpose or aim of this program. So how to apply those tools to analyze the data. So in this module, we would cover the applications of bioinformatics in hematology and serology. So those basically those tools used in blood-related area, and then the bioinformatics in microbiology. So how to analyze sequencing result from bacteria, from virus, which I think probably a hot topic nowadays because of this COVID, a lot of sequence has been generated from virus, from this SARS-CoV-2. So we would have good example of analysis, and the application in general pathology, especially those data related to tumor or cancer, to understand how those mutations, how those genetic problem may link or may cause cancer, and then also how to use the analysis result to trace the treatment, to monitor the process, the development process after treatment. And then we would also cover those pre-implantation and prenatal diagnosis in the lab. So that's also a very common test that's to determine if there's any genetic disorder in during pregnancy in embryo in those babies. And then very important, the last point is the data quality assurance. We would also cover the tools to help you to filter those data to decide the quality of data, and then to identify if the data is good enough for analysis. And after the treatment, we would also be able to identify whether the result is reliable or is it trustable from the tools we use. Because for bioinformatics tool, there are quite a lot of different software for you to use, and whatever you use, it could always generate a result. But whether the result is reliable, whether the result is meaningful, got any real interpretation from those data, then that's a very important issue. So we would learn from this program how to identify whether the result is good, is useful. Well, so the pass mark for each module is 50%, and then students are expected to pass all modules, and then there would be one reassessment chance for the failed module. And then, of course, there's attendance requirement for about 80%. Once the students successfully complete all modules, there would be awards, parchment, postgraduate certificate in bioinformatics for medical laboratory technologists. So in this program, we have invited an experienced panel of teachers who are those academics from local universities who are working on bioinformatics, and then also the professionals from the different labs who are using who are already apply this bioinformatics in their daily data analysis. So they will share their experience. They will teach how to use the tools in processing biological or medical data. Well, we would mainly for module 1 teach in the standard classrooms, and then we also have some practical sessions for you to understand those principles of sequencing technology or of molecular technologies. Well, for module 2 to 4, because we will cover a lot of aspects in computer related issues, so we would deliver in computer lab, so you can just practice immediately after you learn from the teacher. I think for all these programming related area or computer tools, computer software, it's very important to use rather than learn in a traditional classroom by lectures only. And then we definitely will provide e-learning platform to support you, and then provide all the necessary resource, teaching resource during the study. And we would also provide library cards for the students, so students will be able to use online library or use the or just go to the library to get the useful resources, to get like reference books or textbooks. Well, I think the last point is the entry requirements. So who can study how to apply to this program? Well, we would expect the applicants would have a degree in medical laboratory science or in biological or medical related science, so hold a degree in such area, and then also a registered MLT with the HK MLT board. So that's the requirement. And as this program will be taught in English, so we would expect students to meet the minimal requirements of English, either it's IELTS or TOEFL. So that's the requirement. It won't be very difficult. Like basically, if you pass A-level or DSE English at grade E or level 3 and above, you already meet the requirement. Well, you may ask if you are not a registered MLT, or if you are, for example, you are scientists who are working in a lab can you apply? Yeah, if you hold other qualifications, but within 3-year relevant working experience, then we would consider as individual cases. So, so you also can apply, but please submit all the relevant documents to prove your working experience, your qualifications. Yeah, I think that's the end of my presentation. Thank you. Thank you. Yeah.